says switch displays.
Say it again.
A button that says switch displays.
Okay. Oh, I see. As to show the next slide. You've been researching the web.
Maybe it's not.
That's all right. We'll figure it out later. I have 2 more lectures coming up, so we can figure out for them. Thank you. It's out there somewhere. All right. Welcome. I'm Jeremy Nathans. I'm delighted to be here. And today we're going to talk about DNA mutation, damage, and repair. You have the PowerPoint. Are we recording? I think we are. Some technology people were here, and they pushed the recording button. I know nothing. Okay. Thank you. So you all have the PowerPoint, and the PowerPoint both serves as the lecture presentation and also a set of notes. So there's some slides that we won't spend a lot of time on. It just often there's text that kind of lists things for your convenience. So if we skip quickly through a few slides, don't worry about it. They're yours to study. So you might ask, why should we study this? Why devote so much time, a whole lecture to mutation, damage, and repair? And there are various answers. One of them, of course, is that without DNA mutation, there'd be no evolution. We would all be single-celled sea slugs. And we keep evolving, of course, not just on a millions of years time scale, but also on a just weeks, months, years time scale. And I include somatic evolution. And that brings me to the second reason we study it, which is the most impactful somatic evolutionary change is a set of mutations that cause uncontrolled cell growth, which we call cancer. So 1 in 5 people in this room will have medically significant cancer at some point in their lives. 100% of people in this room will have tumors. You already have them. We have multiple tumors. You just look at your skin surface, you can see typically multiple tumors. But there are many more inside. As a medical student, I spent a month doing autopsies. When you do autopsies, you see all kinds of tumors inside of people, most medically insignificant, but again, just changes in the genome that cause the cell to grow uncontrollably. And also, it turns out the genome is devoting a large amount of its information to maintaining the fidelity of itself. So there are a lot of DNA repair proteins. This is sort of a third reason to study this, because the genome is telling us we should by virtue of the head count. A lot of genes are devoted to sensing DNA damage, to responding to it, to repairing it, and dealing with the consequences. Okay, so first a big thank you to Jeff Corden. I took off from his lectures when I began this one. And here's some learning objectives. You can read those at your leisure. And here's just a single-page summary of what we're going to talk about today. Again, read it at your leisure. And then here's an outline. Okay, so we're going to talk about 5 topics. And the first is DNA mutation and DNA damage. Well, even independent of external forces, external insults like radiation or chemical mutagens, there is an intrinsic challenge to maintaining the fidelity of a DNA sequence, because there are certain chemical instabilities which nature just can't get around. Here's one of them. It's called tautomerization. And on the left, if I have a laser here, yeah. Okay, so here's a normal AT base pair, thymine on the left, adenine on the right, and they're shown in their most common, most energetically favorable configurations. But it turns out there is a version of adenine which looks like this. It's called the amino version, in which this nitrogen here in the middle of the ring has picked up a hydrogen, and the double bonds have flipped around. You can see the arrangement of double bonds in the six-membered ring are different. And there's a double bond going up to the amino group that sticks out, and it's lost one of its protons, lost a hydrogen. And this reverses the hydrogen bonding acceptor-donor characteristics of this nitrogen and that nitrogen. Now the one in the middle is a hydrogen bond donor, the one at the top is an acceptor, and that means that the amino version of adenine can base pair with cytosine, which it shouldn't be doing, instead of thymine. Now, this is a minor form. Minor means we now know less than 1 part in 10,000 at any moment in time. But remember, the human genome has 3 billion base pairs, the haploid genome. So that's still a lot of adenines that would be in the amino form at any moment in time. And if that moment in time happened to be when DNA polymerase is replicating with this base as the template, then it's going to put a cytosine opposite this adenine. Of course, when it reverts back to its more common form, there'll be a mismatch, an AC mismatch, which could be recognized by the DNA repair enzymes. So this is not good, but it's not as bad as it might be because of, and we'll get to the enzymes in a few minutes, because of the enzymes that are constantly patrolling DNA looking for mismatches. Here's another spontaneous event that is unavoidable, and this is depurination. So if we look at the deoxyribose, the backbone here joined to a base, and it's particularly common with purines, that is A and G, as opposed to pyrimidine, C and T. But guanine, shown here, one of the purines, has a linkage to the base through this nitrogen, which is not, as all linkages are, 100% absolutely stable. It has some rate of hydrolysis. This carbon can be attacked by a water molecule, and the stability is on the order, half-life is on the order of 1,000 years. That's not too bad, but remember, we got 3 billion bases, or base pairs. So at for any cell, say summed over a day, there are thousands of these events, depurination events, where the base just drops off, and now you have a depurinated sugar. And that, again, is recognized by enzymes that are looking for this sort of error. So it's not as bad as it could be. And then finally, here's one more. This is a spontaneous reaction. It's the hydrolytic deamination of cytosine. So this carbon here, where the nitrogen is bonded, can be attacked by a water molecule. It kicks out an ammonia. You get a carbonyl in its place. And this is uracil. So remember, DNA has thymine. RNA has uracil. They base pair the same as each other. They differ because thymine has a methyl group sticking out here. Uracil doesn't. But if a cytosine deaminates and becomes a uracil, now we have a U, which is in the DNA, and if that replicates and the U is the template, there'll be an A placed opposite it. So again, the fidelity machinery will want to catch this before the next round of replication. Now let's talk not about spontaneous events, but about environmentally induced damage. And there are various ways that happens. One of them is via light absorption. Ultraviolet radiation can induce two especially common adducts. These are a cyclobutane. This should be an A up here. It's not butene, it's butane. Cyclobutane dimer. So when there are two different, when there are two pyrimidines adjacent on the same strand, this isn't sort of drawn to scale, but they can, thanks to UV irradiation, be joined by this four-membered ring. Remember from organic chemistry, four-membered rings are really not particularly stable. In fact, they're highly strained. But given their close proximity, that's what happens. Or there can be a single bond linking them. So either of these kinds of adducts. And that is trouble for the double helix. Here's another environmental insult, not radiation, but chemicals. So various planar conjugated ring systems like this one, ethidium bromide, which you've probably worked with, stains DNA, can interact with DNA by intercalating between adjacent base pairs. So remember, the planar surfaces of the bases are fairly hydrophobic. and exclude water. And another hydrophobic molecule, and this surface is certainly hydrophobic, is looking basically to escape the aqueous environment. And so by hopping in between the adjacent bases, that's a low energy state. And so it slides in. The way ethidium bromide works, the reason you can look at DNA on a light box and see the fluorescence is because the UV is absorbed by the DNA, it's absorbed by the bases, and that UV energy is transmitted in a non-radiative reaction to the ethidium, which then fluoresces. And it's a very efficient non-radiative transfer of energy. But the problem with this is that if the ethidium, from the DNA's point of view, if the ethidium hops in here, it moves the bases further apart from each other, and then during DNA replication, that can be a site at which DNA polymerase will make a mistake, thinking that that spacing is the right spacing, and add a base, for example, where a base shouldn't be. So actually, it turns out it can add or subtract, depending on whether that's the template strand or the newly synthesized strand that's happening to. And so intercalating agents like this give single nucleotide insertions or deletions as their mutations. Here's another environmental insult. This is alkylation, and it can be a methyl group or an ethyl group, some alkyl group, which can be added to especially the oxygen at the 6th position of guanine. So here's the oxygen. This is a site for potential alkylation, and here's the product shown here with the alkyl group sitting on it. That also changes the base pairing properties. It makes it more likely to pair with a T. Of course, G should be pairing with C, but the O6 methyl G pairs with a T. So again, you can see during replication, that would be mutagenic. Of course, in the lab, we use this to our advantage. You don't want to be drinking this stuff, but we use it for mutagenesis screens. Ethyl methane sulfonate is the favorite. The ethyl group is the one that can be transferred, and so you get an ethylated guanine. Now, people have been studying mutagenesis for a long time, initially just by classical genetic methods, and then, of course, with more molecular methods more recently. And here is the premier system for studying mutagenesis. It's the lac repressor system. So just a little background, in E. coli, there's an operon, the lac operon, which mediates the utilization of lactose as a carbon source. Lactose is glucose and galactose joined together. It's a disaccharide. And so this operon encodes proteins for a protein for import of lactose, and then also a protein called beta-galactosidase, which splits the lactose in the middle into its glucose and galactose components. And the whole thing is under control of a repressor called LAC-I, which is actually just adjacent, just upstream of the operon of genes, LAC-O, LAC-Z, LAC-Y, LAC-A. Here's the promoter P. And LAC-I encodes a protein, the lac repressor, which binds, this isn't drawn quite right, it actually binds closer to the promoter. But, oh, I guess LAC-O is the, sorry, it's the operator. Yeah, this is right. So LAC-O is the operator. So it binds just downstream of the promoter, and when the repressor binds there, it inhibits transcription. So it's what you might expect. You don't want to be making all of these gene products when there's no lactose around. That would be a waste of energy for the cell. And so the repressor keeps it turned off. But when lactose is present, it binds the repressor and changes its conformation so that it can no longer bind DNA, and transcription is initiated to produce the LAC-Z, LAC-Y, LAC-A gene products. And here they are, the 3 proteins. Now, one more piece of data. There's a chemical substrate called X-gal, which is colorless, and which you can just mix into the agar of your plate. So you pour a Petri plate, it's got the usual L broth, but it also has X-gal in it. X-gal is a substrate for cleavage by the beta-galactosidase enzyme, and the product of cleavage is an insoluble blue compound, which accumulates in the colony. So I think you can see on this plate, some of the colonies are blue, they're also a little bit smaller. I think they don't like having this blue precipitate in them. And others are white. And the white ones are the ones that do not make beta-galactosidase. And so, for example, in this experiment, they have an intact lac repressor. And there's no, I should say, there's no lactose in this plate. So this operon should stay repressed. So they have an intact repressor. They're white. They don't make any beta-galactosidase. They don't, in fact, make any of these gene products. But then there are some colonies that have a mutant lac repressor, so it cannot repress, in which case the operon is expressed, and there's plenty of beta-galactosidase, and therefore those colonies turn blue. So this is great. You can just look at the plate and immediately tell which colony has an intact repressor and which one doesn't, and you can easily pick out 1 in 1,000. So that has been used to study mutagenesis because you can apply a mutagen or irradiation, whatever you want, and then look for the blue colonies, the ones where you've knocked out the repressor, and then recover the repressor and gene and sequence it. And the first thing that came out of these studies, and the hero of the stories is a fellow named Jeff Miller at UCLA, is that mutations are highly non-random. You might have thought, oh, every base in the DNA is just as mutable as any other base. Mutations can happen anywhere. What's the big deal? That's totally wrong. Mutations occur in a very distinctive pattern, both spontaneous mutations and induced mutations, and they occur highly non-randomly. So here's just a sampling of the lac repressor gene from nucleotide 1 to 350. And here in every little square or circle is a single mutation. So just a collection of lac repressor mutants, sequence them all, see what you get. And I think you can appreciate that there's one place where there's a whole pile of mutations shown as red squares. And actually at the same place there's another pile of mutations shown as blue squares. All the red ones are identical to each other, but they're independent events. All the blue ones are identical to each other, but they are independent events. And then there are a series of deletions, some of which are recurrent. They keep happening over and over again, and in various places. Okay, so what if you look at the sequence, what's going on at these various places? Well, it turns out that the place with the most common mutations is a small region of repetitive sequence. So the wild type sequence is CTGG, 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 so 3 copies. And the most common mutation is to amplify it from 3 copies to 4, which of course, since it's not a multiple of 3 nucleotides, introduces a frame shift. So of course that destroys the protein's function. And the second most common was the loss of 1 copy. Again, a frame shift because it's 4 nucleotides. And here we are down with just 2 copies. This kind of error happens during DNA replication. There's a slippage of either the template strand or the newly synthesized strand. And this is exactly the kind of mutation that is most common in us, in humans, and in pretty much every organism on the face of the earth. That is the slippages of these repetitive sequences. In us, these things tend to occur about 1 every 1,000 generations at any particular place when there's a set of repeated sequences. How about the deletions? Well, interestingly, the deletions also tell us something about repetitive sequences. The most common deletions, the recurrent deletions, have repeats of the same sequence at their termini. So for example, here's a common one. It's 75 bases long, and there's a sequence GTG, GTG, AA, which is repeated at the other end. And these two, basically, there's a recombination event between these two, or a DNA polymerase slippage. We're not really sure which it is, but the resulting deletion leaves you with just 1 copy of the repeat and nothing in between. Everything in between has been deleted. And again, in us, that's exactly the structure of the most common gene copy number variants, so-called copy number variants, deletions or duplications. There are repeated sequences. The region in between either gets completely removed and just 1 repeat is left, or the whole thing gets duplicated by a recombination event, and now there are 2 identical repeats, 2 identical units with the repetitive sequence, the unique DNA, repetitive sequence, unique DNA, and the repetitive sequence. So one of the nice things about studying mutation and repair and DNA damage is that the themes tend to translate across all organisms. I mean, DNA is basically DNA, whether it's ours or E. coli, and the strategies for repair and the mechanisms of damage are generally conserved. Now, even at the level of point mutations, there is a lot of non-randomness. So if we look at the most common point mutations, it turns out they occur where there's a cytosine, and it's a particular kind of cytosine. So here's a plot just showing along the length of the LAC-I gene the likelihood of getting a mutation at any particular base. These are single base changes, and you can see that there are a series of very tall peaks. They have little asterisks because these are the places where there is a cytosine that's methylated. So E. coli, like us, puts methyl groups on a subset of its cytosines. And those base pairs, of course, those are GC base pairs. When they're mutated, they invariably become AT base pairs. So C becomes T, G becomes A. And why is that? Because, as I mentioned earlier, if there's a cytosine and it gets deaminated, you get a uracil. But the uracil, being distinct from thymine, is recognized by an enzyme system. It's called, as you might have guessed, uracil N-glycosidase. It's an enzyme that when it sees a uracil in the double helix, it cuts it out. It cuts it off the deoxyribose base, and that leaves a site that is lacking a base. And then that can be recognized by yet other enzymes. But I think you can appreciate if there's a methyl cytosine, it's 5-methylcytosine, this modification, which is made, I should say, after the double helix has been synthesized. It's not part of the precursor deoxynucleotide triphosphates. They're enzymes that, looking at double helical DNA, will put a methyl group on cytosines. That methyl group means that when this gets deaminated, we don't get a uracil, we get a thymine. This is dangerous because the thymine now is indistinguishable from a normal thymine. So there's no way for this uracil N-glycosidase system to recognize it and cut it out. The only way to recognize it is as a mismatch. It's now a thymine base paired with a G, with TG base pair. That's an erroneous base pair, so that can be recognized. But the recognition has to then know which way to correct it. And we'll get to that in a minute, that how the system knows which strand is the right one and which is the wrong one if the base pairing is incorrect. But the result is it doesn't always get the correction done appropriately or in time. And so cytosines that are methylated will tend to lead to GC to AT mutations. And it's the same in us, exactly the same in us. Those are the most common point mutations in humans. Okay, now you might be wondering, since we've talked about E. coli and the lac repressor, what's the analog system for studying damage and mutagenesis in, for example, human cells? And here's one trick which is very simple and kind of clever, and that is basically to stick with the lac system, but simply put the DNA into mammalian cells, do the mutagenesis, and then allow time for the repair to happen in the mammalian cell, and then recover the DNA back into E. coli and analyze its sequence. So this is done here, and what you see is, for example, if you treat mammalian cells, this happens to be human 293 cells in culture, that have been transiently transfected with a plasmid that encodes the lac repressor, and then wait a couple days, and here they've been treated with ethyl methane sulfonate, put them back in E. coli, and then look for those plasmids which are lac mutants. So just to give you a little detail here, the E. coli recipient has no wild type repressor gene. So it needs to get the repressor gene from the plasmid. And the repressor gene from the plasmid can either be wild type functioning or mutant non-functioning. And if it's non-functional, then that E. coli will turn the X-gal blue. And so you can see which colony it is and you can sequence it. And when you pick several hundred of them and sequence them, what you see is this very beautiful pattern. All the mutations are, or almost all, not all, almost all are GC to AT base pair changes, not GC to TA, except for one exception, not AT to TA, not AT to CG, and so on. That is, the mutagen has a very definite specificity in terms of what changes it produces, and that accords with what we know about ethyl methane sulfonate as an alkylating agent. Now, just to generalize the testing a little more, it would be nice to have a test that could, in a single, simple, inexpensive, rapid way, show you the mutagenic potency of any kind of treatment, either a chemical or radiation treatment. And it could be used, of course, for monitoring environmental samples. And this is the Ames test, invented by Bruce Ames. And the idea is shown here. You take a rat liver extract. Here's just a little detail, but I'll mention it. Why do we take a rat liver extract? Because the liver is a detoxifying organ. So when you eat, McDonald's hamburgers, whatever, with all the nasty stuff in them, the food that you've ingested from your GI tract has a first transit through the liver, and the liver enzymes, and there's an enzyme system called P450, cytochrome P450, has a chance to detoxify whatever is in there. So if we were to mix our potential mutagen with an extract of liver, we're actually then giving the mutagen an opportunity to get transformed into something that's a little more like what would happen in a living organism. And I should say that the detoxifying system, although it generally does a good job at getting rid of bad stuff, it also has the unfortunate property that it not infrequently modifies the mutagenic compound to make it even more mutagenic, a bad design, as it were. And we'll see that in a minute. So we take the liver extract, we mix it with a salmonella strain. It could have been E. coli, but Ames happened to use salmonella, that requires histidine. It cannot make its own histidine. You know, most bacteria can make most of their own amino acids. We can't make all of our amino acids, but bacteria typically make all of their amino acids. So what Ames has done is he's created a set of histidine, so-called auxotrophs, bacteria that require histidine to be added to the plate in order to form a colony. And he has a whole collection of them whose sequences that cause auxotrophy are known. They've been sequenced, and we know that some are insertions, some are deletions. There are various point mutations of all different kinds. And each member of this collection is mixed in a tube with the liver extract and then with the potential mutagen, or not, or a control, and plated on plates that lack histidine. So the question is, will colonies form? And if a mutagen can revert the mutation, so let's say it was a C to T mutation that was in this particular mutant strain. If the mutagen is capable of making T go back to C, then it will convert the mutant histidine auxotrophy marker to a wild type one. And then colonies will grow on the plate. And all you have to do is count the colonies to see the potency of the mutagen. Whereas the control plate has very few colonies because spontaneous reversion is rare. Here's Bruce Ames. And here's just an example of one of the nastier compounds that's out there. This is aflatoxin. It's made by molds. These molds grow in all kinds of plants. Here it is on an ear of corn. If you see an ear of corn like this, do not eat it. Aflatoxin, shown here, is nasty enough by itself, but it turns out when it gets activated by the cytochrome P450 enzymes in our livers, it gets even nastier. And this is a major source of hepatocellular carcinoma, liver cancer, throughout the world. Okay, so just a little summary here. DNA damage can occur through chemical reactions inherent in DNA structure, like tautomerization, depurination, deamination, chemical reactions induced by metabolic products like alkylating agents and oxidizing agents, and environmental factors like radiation and genotoxic chemicals. Okay, so here's a little question just to make sure we're awake here. Which of the following statements is true? DNA damage and mutation are the same thing. Any hands? No, because you can have damage, but it could be corrected, right? Not necessarily going to be a mutation. DNA damage occurs all the time and in all cells. Yeah, you got it, all the time, like right now in us. And DNA damage always leads to mutation. No, that's definitely wrong. Okay, in fact, it rarely does. Okay, voila. Okay, now for part two, repair of incorrect or damaged bases. Okay, so there are various repair systems. Let's just look at a few. Here's one in which the UV irradiation product, this cyclobutane ring or this 6,4 adduct, can be reversed by a photochemical reversion. So it was induced by light. It can be reversed by light. And there's an enzyme that uses this light harvesting cofactor, which actually binds these adducts. It recognizes them in DNA, and using visible light energy reverses the reaction. Sadly, we don't have that in our genome. So bacteria have it, but we don't. So good reason to stay out of the light, especially if you have minimal pigmentation. Here's another way of reversing damage. This is just shown sort of schematically here with the damage shown as these little diamonds. This is for alkylation. It turns out I won't call it an enzyme, it's really a suicide protein. It's a protein that recognizes the alkylated base and then attacks the alkyl group and picks it up onto the protein itself taking it off the DNA. That's a one-use protein. Once the protein has the alkyl group, it's basically not good for anything else. It gets thrown away. But it has saved the DNA from the alkylation damage. Interestingly, the same protein also controls its own transcription. And so in the alkylated form, that is the form you get when there's a lot of alkylation damage in the cell, it actually activates its own transcription to make more of itself. That's kind of cute, because that's at a time when the cell needs more of that protein. Now here's just a summary of what we're going to talk about for the next few minutes, various ways of correcting DNA damage, and this is true in bacteria, it's true in plants, it's true in us. So I'll just go over them briefly. One is base excision repair. So this is a little small, I hope you can see it in the back. Basically, you can get rid of a damaged base with an N-glycosylase, an enzyme that cuts it off the sugar, and then that site with no base is recognized by another series of enzymes which make an incision in the DNA, and it's repaired by DNA polymerase. Nucleotide, and it really should be oligonucleotide excision repair, because it excises a little chunk of single-stranded DNA, an oligonucleotide, maybe 10 or 20 bases long, is just another strategy. If there's DNA damage and it's on one strand, you can make an incision upstream, you can make an incision downstream. This little oligonucleotide is then removed, and the single strand is resealed, or resynthesized by DNA polymerase, and then resealed by DNA ligase. Now here's one that's kind of a last resort, translesion synthesis. So this is especially useful during DNA replication. If there's a lesion and the replicative polymerase, which is this very complex machine and rather intolerant of DNA modifications, erroneous sort of modifications, chemical, for example, modifications, the replicative polymerase will stall if it hits one of those things. And if the cell is basically trying to replicate its DNA, it doesn't have time for this sort of stalling business. It needs to keep the show on the road. And so what it does is it uses a so-called bypass polymerase, and there are a whole series of them, which are error-prone polymerases. They just synthesize across the modified base willy-nilly, often introducing an incorrect base during that synthesis. So they're mutagenic polymerases in that respect, but at least they get across the modification, and then the replicative polymerase can take over. And then finally, another totally different strategy is to use information from a duplicate double helix. For example, a separate chromosome. In us, of course, most of the genome is present at 2 copies. This is true except for the X and the Y in males. It's present at 2 copies, and so the other copy is around as sort of a backup. And if information can be donated from the good copy to fix the bad copy, that's another strategy. Okay, so let's just, I'm not advancing here. Let's try an escape. Let's see. I think I need technical assistance. Is there technical assistance? This could be a short lecture. Well, maybe we'll turn the thing off. Is Phil in the back there? No, he's abandoned us. Any thoughts? Anybody major in computer science out there? Oh, you did it.
There you
go. A genius. Round of applause. All right. So here, I'm just going to show you now in a little more detail these separate mechanisms. And these are basically your notes, so I'm not going to go through every little bit here. So this is base excision repair. There's a damaged base. The N-glycosidase clips it out. Now there's an purinic site that's recognized by this nuclease, makes a cut, DNA polymerase fixes it up. Interestingly, if you actually look at the crystal structure, these glycosylases actually swivel the base out of the helix. It's kind of cute. They have a good look at it all by itself out there. They decide it doesn't look right, and they snip it off. here's the oligonucleotide, often called nucleotide excision repair pathway. A little more complicated, a set of enzymes in E. coli, UVRA and B, another UVRA on this side. They see this distortion in the helix. UVRC comes along. It makes nicks here and here on either side. This little chunk gets removed, and then DNA polymerase seals it up, synthesizes and seals it up with this green segment here. We have essentially the same system. In general, we have more proteins doing the same thing that fewer proteins do in E. coli. It turns out that there are people with mutations in the proteins that do this sort of nucleotide excision repair, NER. They have various disorders, all of which are unified by a very high frequency of skin cancer, because there are lots of UV-induced mutations which are not being appropriately repaired. And the disorders are xeroderma pigmentosum, Cockayne syndrome, and trichothiodystrophy. But in terms of the enzymology, it's essentially the same reaction. Snip out, here are the scissors, snip out a little bit of DNA and resynthesize. Now here's a mechanism that I've sort of alluded to earlier, which is fascinating. It's post-replicative repair. So if a mistake has been made, for example, DNA polymerase incorporates the wrong base, there's a telomerization or just a mispairing, can you, after the fact, fix it? And very early on, it was clear there must be a mechanism that does this. So the answer is yes, you can go back and fix it, because we know that the DNA polymerase that E. coli uses makes only about, makes about one error in about 10 to the 7 bases. Okay, one error in 10 million bases. That's pretty good, but we know that in E. coli, the mutation rate is only 1 in 10 to the 9, 1 in a billion. So 99 out of 100 of these mistakes must get fixed somehow. And the answer is there's a post-replicative system. It's also true in us, true in plants, true in fungi. Another sort of way to look at the newly synthesized DNA with a second pass to see if it's okay. And in E. coli, this is governed by methylation. It turns out that when DNA is first replicated, because the deoxynucleotide triphosphate pools do not have methylated nucleotides in them, all of the new DNA is unmethylated. But, and so here, here's the old DNA, and in this case, it's for E. coli, it's methylation on adenine, and it happens at these sites, GATC, and I think you can see on the other strand, it goes the same way, GATC, so it's a palindrome. The two A's carry a methyl group, and when DNA is newly replicated, the initial product strand doesn't have the methyl group. And there is a methylation called DAM methylation. We often refer to it affectionately as DAMN methylation, because when you're trying to do a restriction digest and you forget about this and your restriction site happens to encompass this sequence and your enzyme doesn't cut because it doesn't like methyl groups, you realize it after you've run your gel that there's no cutting, and you say, damn, methylation. Okay, so after a few minutes, the newly synthesized DNA is doubly methylated, because this enzyme will find the hemimethylated sites, which are its preferred substrate, and put a methyl group on that adenine. So it restores the original arrangement. But during that short time interval, there's an opportunity for this fail-safe mechanism to look for mismatches, and using the fact that the old strand has the methyl group and the new strand doesn't, know which strand is the mistake, that is the new synthesis, and which is presumably the correct one, the old one. Okay, and here's Paul Modrich, who discovered this system at his lab in Duke University. And here's the way it works. It turns out, of course, that the mutation shown here is this GT mismatch. Isn't, of course, right next to necessarily one of those methylated adenines. That methylated adenine could be 100 or a few hundred bases away. So how does the place where the correction happens know which strand is the old and which is the new strand if the mark for old and new is so far away? And the answer is a series of sliding clamps. So this is shown in very schematic form, but this is a hexamer of a protein that literally forms a bracelet around DNA. And once it's on the DNA and it's put on in a way that is asymmetric, it can slide along the DNA, preserving the information, preserving the mark of which strand was the old and which was the new. I'd say this is put on at the, or it's set asymmetrically at the place where the asymmetric methylation is, and then the enzymes that do the repairing, MUT-S, MUT for mutation, S and L, engage this ring also in an asymmetric way. So they know which way to approach the DNA based on how the ring was sitting on the DNA, which can slide, as shown here, can slide back and forth, but always maintaining its orientation with respect to new and old strands. So that's the way the memory of what is new and what is old is maintained up to the point where the mutation is identified. I think we can skip that one. Okay, here's just the translesion synthesis. This is that mechanism of last resort that I mentioned earlier. Here we have a DNA modification that would block the replicative polymerase, and the translesion repair polymerase hops on just for this little interval to synthesize across that place and then hands it off back to the replicative polymerase. Okay, and as I mentioned, this is highly mutagenic. The main thing from this slide is that the error rates are of the order of 1 in 1,000, very high mutation rate, by having this error-prone polymerase take over the job for this brief time. Finally, let me just mention just one of the many repair machines in all organisms, us and bacteria. This happens to be the set of proteins in humans, and it's quite a complicated set, but there's a kind of DNA modification which involves cross-linking between the two strands. Now, that's really trouble, because of course the DNA replication fork, when it gets to that place, it can't pull the strands apart. They're covalently linked together. And so, as you might guess, if there's a lesion, there's also a repair system. And here are the little balls indicate all the proteins. It's incredibly complex. It recognizes the strand joining and breaks it apart. And I should just say this is also associated with disease in humans. It's called Fanconi anemia. It's a terrible thing to have because it turns out these sorts of crosslinks happen all the time in all cells, and these people have multi-organ dysfunction. They typically die before age 20. Okay, so here's a summary. Mismatch or bulky adducts are recognized by proteins that sense changes in DNA structure or flexibility. Specific pathways have evolved to repair different types of damage. In the absence of repair, error-prone DNA polymerases replicate across the damage base. Okay, so now topic 3. And maybe at this point, let's just take a little 1 or 2 minute break. Just stand up and stretch, because it's an hour-and-a-half lecture. A stretch will be good. But try not to leave the lecture hall unless it's an emergency. Okay, let's do the second half. Okay, so now we're going to talk about repair of double-strand breaks. We've up till now talked about single-strand adducts and single-strand problems, but what if there's a break across both strands of the helix? So this can happen with, for example, very high-energy radiation, like X-rays or gamma rays. There are 2 ways to put this together. One is called non-homologous end joining, and this is basically an act of desperation. Just take the 2 fragments and smash them back together again, because having the 2 ends just floating off separate from each other is really bad news for example, DNA replication. So this mechanism generally gives an imprecise join. Very often there are bases missing as a result. We'll look at it in a little more detail in a minute. It's the thing that we use when we do CRISPR editing. So the typical CRISPR mutagenesis gives, there's a cut, a double-strand cut across the helix, and then that's repaired in this error-prone fashion. But there's another way which is a little more tidy, and that's to use the information in the other homologue. If, for example, there's an organism with 2 chromosomes like us, or, for example, if it's a bacterium and this double-strand break happened to occur when the bacterial genome was partly replicated. And so the other copy, the newly synthesized copy, is available with correct information. To use that correct information to put these 2 pieces back together again correctly. So this is the proverbial Humpty Dumpty getting put back together again. And this is just shown in cartoon form what that would look like with a precise joint using the information from the other homologue. Okay, so if we look at non-homologous end joining, the sloppy, desperate system, here's sort of 2 ways that it can happen. You can get the initial cut, and then typically nucleases, which are abundant in cells, will degrade a little bit back from the ends. And when these 2 pieces get ligated back together again, often nucleotides are missing. So 2, 3, 4, 5 nucleotides are gone. Another way this can happen is there can be degradation of one of the 2 strands because there are single-stranded nucleases around. And if that happens and the overhang, this is sort of a random event, but if the overhang happens to have homology, you can see the GAT on the upper strand is can pair, the TGAC, pardon me, on the upper strand, compare with the ACTG on the lower strand, as shown here, with some single strands hanging out in the breeze, then these 2 can pair with each other. The stuff that's single-stranded gets degraded away, and this gets ligated together. Again, we've caused a deletion here. So this is still sort of sloppy. This is the non-homologous end joining slop that invariably causes mutations, or almost invariably. But now here's the more tidy way to do it, using the other homologue. And this will be a recurrent theme in using another chromosome to basically donate information. It's a series of steps, and the only way information can be donated is via Watson-Crick base pairing, but the hydrogen bonding base pairing. And so how do you get that information over to the chromosome that needs it? Well, you have to resect one of the strands. Usually typically happens on both sides. So now we have single-stranded DNA. That strand can invade the good double helix. You might be wondering, how does it find the correct sequence? After all, this chromosome is just a little part of the vast genome, and this little part of the chromosome is just a small part of this much larger set of sequences. It turns out this is one of the sort of miracles of enzymology. They're enzymes that catalyze this sort of strand invasion. They pull these strands apart. They allow this single-strand DNA to basically sniff around looking for Watson-Crick base pairing. And if it finds it, it will then form this sort of semi-stable base paired region. I should say it's the same sort of search challenge for the CRISPR system. The CRISPR uses a RNA guide to find its target in the genome. But again, that's a search problem where it has to search through the entire genome. But once that's happened, I think you can see now how information can be transferred. For example, if there were a mismatch, there isn't in this example, but we'll see one in a minute, that mismatch could be corrected. In this case, what we're asking for is synthesis from this 3' end by DNA polymerase, which extends the invaded region further. And having extended it, now these base pairs can be broken. This newly synthesized single strand can go back to its original chromosome, but now it's been extended, and it includes the region that overlaps the break. And so when it base pairs on the distal side of the break with the single-stranded DNA that was this region here, it now closes the gap, and the closure is perfect in terms of information. Nothing has been lost at this point, and the gaps can be sealed by DNA polymerase, by synthesis by DNA polymerase, and then by ligase. So now we've got ourselves back to the original arrangement with, in green, the information that was donated from the red good copy. Okay, that's the way to finesse it and correct it perfectly. And here's just a slide just showing one of the strand exchange proteins. This happens to be the E. coli version called REC-A. It binds single-stranded DNA, and it catalyzes this invasion of another helix by that single-stranded DNA. So this doesn't happen spontaneously. You can't just mix two pieces of DNA in a test tube and get this to happen. It won't happen. You need enzymes to do it, and it needs ATP. And again, a disease connection. Turns out that the breast cancer susceptibility gene, BRCA1, a tumor suppressor gene, which, if mutated, vastly increases the risk of breast and ovarian cancer and other cancers, is part of the machinery that does this in us. This is just showing that in detail. After the resection, there's a protein like the REC-A protein. It's called RAD51 in us, and it mediates this change, this invasion. But all of this requires the BRCA1 recognition of the double-strand break. Okay, so here's a summary. I don't have to read this at this point. You can read it on your own. And now let's talk about topic 4, damage sensing and inducible repair proteins. So as you might guess, sensing damage is important because the repair enzymes, repair systems, can be induced if they're needed. So under normal circumstances, there's sort of a basal level of these repair proteins around, but if there's a lot of damage in the cell, you'll want more of those enzymes, and in fact, their synthesis is regulated in just that way. And here's how it works in E. coli. We have a similar system that we'll talk about in a minute. This was discovered by Evelyn Witkin in the 1940s. She died just a couple years ago. In fact, I have a picture of her. I'll just show a picture. This is Evelyn Witkin, actually at age 100. Amazing woman. She was a professor at Rutgers. And I'll just say she was your age when she discovered it. She found this system called SOS, Save Our Ship. This is the help signal for a sinking ship. And this is sort of the reason that name was given. It's the analog for an E. coli that's in trouble. Basically, the REC-A protein, the one that mediates strand invasion, turns out to be, amazingly, the sensor also for DNA damage. So it does double duty. And the reason is because normally in a cell, any bacterium, there's almost no single-stranded DNA present. It's a transient intermediate during DNA replication, but there's very, very little of it. But if there's DNA damage, then the replication forks will be stalling. For example, the fork which is moving in the contiguous direction, the contiguous synthesis fork may continue, but the lagging strand may be blocked and there'll be single strands accumulating there. There's all sorts of ways that single-strand DNA accumulates secondary to DNA damage, and therefore the REC-A protein finds its substrate, single-strand DNA, it binds that substrate, and it changes its conformation. And that altered conformation turns out to reveal a protease site. It is not just a DNA binding protein, not just an ATPase, not just a strand invasion protein, but it is also a protease, but it's a latent protease. And when that protease site is revealed by REC-A's interaction with single-strand DNA, it's a very specific protease. It cleaves a repressor called LEX-A, which is shown as these two little balls joined together. So LEX-A has two domains. It cleaves between the two domains. The two domains now separated destroy the function of LEX-A. And what did LEX-A do normally? LEX-A, as a repressor, bound to the promoter regions of the DNA repair genes and kept their expression at a low level. And when LEX-A is cleaved, it can no longer bind DNA with high affinity, and now those genes are expressed, and so the various repair proteins, and there are about 50 to 100 of them in E. coli, are induced, and those and here are some of them, UVR-A, B, D, there are a whole long list of them. And then when DNA is correct, when the DNA is repaired and the cell returns to its original state and there's very little single-stranded DNA, then the REC-A protease activity is no longer there. The LEX-A protein accumulates in its normal native form, and it goes back to repressing the repair genes. Okay, so discovered by Witkin. And here's just an example to show how rapid this is. This is an amazing system. This is just monitoring the induction of a particular gene, SFIA, just happens to be involved in arresting the cell cycle because you want the cells to stop replication, just halt, the whole business and fix their damaged DNA before resuming replication. So not surprising that this one would be induced. And here is time in minutes on the horizontal axis. And here's the level of induction in arbitrary units on the vertical axis. And here are different doses of UV radiation, either 0, the bottom set of data points, 0.8 joules per meter squared, the next set, 1.8 joules, 5 joules. And you can see within 10 minutes, this gene is already starting to get induced. So this system is a very rapid response system. Now we have a much more complex DNA damage response system. I won't go into any of the details. Mariki Leho in radiation oncology here is the local expert. But the logic is the same. That is, there's a series of sensors. In our case, they are feeding into 2 very large protein kinases, ATM and ATR. They sort of integrate all the damage, and then their signals feed out to a series of damage responses. And again, I won't go into all the details. This could be a whole course, but it stops the cell cycle. Cells stop dividing. They got to fix their DNA first. Regulates the genes involved in the DNA damage response, DDR. If it's really bad, the cell commits suicide because there's nothing to do to save itself. And then there's a whole series of stress response genes that also come up. Here's just another sort of theme that is basically the same as bacteria, but the player happens to be different. This is recognition of single-strand breaks and single-strand DNA by a protein called PARP, poly-ADP ribose polymerase. This is famous because there's an anti-cancer drug now that targets this pathway. And PARP basically does sort of like what REC-A does. It recognizes single-strand DNA as a sensor, but the chemistry is a little different. It forms these long chains of poly-ADP ribose. It's kind of weird, but it ligates them onto itself and onto other proteins. And that's the signal that it's time to repair these single-strand breaks. And then when they're repaired, there's another enzyme called PARG, poly-ADP ribose glycohydrolase, which takes off these little pink balls, the poly-ADP ribose, and restores the system back to its ground state. Okay, now the final topic, homologous recombination and its role in DNA repair. So we looked at this a little bit with respect to double-strand breaks, the way you could use a good copy to fix a broken bad copy. And let's just extend that to look at how you can use that to fix point mutations, but also then how it's used for recombination between chromosomes. So here it is schematically. Let's say we have genes A, B, and C, and they come in either uppercase or lowercase flavors. And here are the 2 chromosome copies, so the black chromosome and the red chromosome. And you can use the information from, let's say the red one is the good copy, to fix an error, let's say, or just a change in allele, even if it wasn't an error, in the black copy. And so, for example, the product of that would be now a little b version of this middle gene in the black chromosome that was donated by the red chromosome. Or you can do a recombination event here right in the middle in which the 2 flanks have been exchanged. And all organisms have this kind of mechanism. Now, it proceeds by what's called a holiday junction, not like Christmas holiday, but there's a person named Holiday, actually 2 L's here. And in the 1960s, he thought of this sort of conceptual way of looking at it. And here's how it works. So I'm just going to walk you through it. So here are the 2 chromosomes. We get a strand invasion as the first step. So from the black chromosome, a single strand has invaded the red chromosome. It's pushed out this single strand out of the way, and here it is now forming a base pair with the lower strand. And you could also imagine then a cut across this region on the right. Can everyone see this little dot? It's kind of tiny. You can see it way in the back? Okay, good. So you could imagine a cut here as a next event, and then the nicks could be sealed up. And if you did that, you would get this intermediate with a single crossover, and this is the holiday intermediate, and a little bit of DNA that's already been exchanged between the two. Okay, now follow me here. Imagine this thing is sort of free-floating in space, and we take the bottom chromosome, little a to little b, and we just invert it. We take it and we flip it around. So now the A side is on the right and the B side is on the left. Here it is. If we do that, we've unwound the crossover in the middle, and we have this structure in the middle. So this is the same thing. We've just flipped around the chromosome. Okay, that's okay. We can do that. And now, if I just redraw it up at the on the right here, up here on the right, but now I bend up the big A and big B side, so they're bent up at 45-degree angles, and I bend down the little B and little A sides, so they're bent down at 45-degree angles. Then it looks like I've drawn, just as I've drawn them here, and in addition, you can draw a scissors cut, either horizontally or vertically. And these are the two ways you could resolve this structure. That is, by enzymatically cleaving and then ligating. So if you do it with a horizontal cut, then what are the products? Well, the A and B arms are still the original black, black, or red, red arms, and there's just this exchange of a little bit of DNA in the middle, either this single red strand or single black strand. And if that exchange region had a mismatch, either because these chromosomes, either one suffered a mutagenic event, or they just happen to have sequence differences between them, those mismatches can now be corrected by the mismatch repair system to create, on either of those, either a red-red or a black-black, a red-red or a black-black. If we break it vertically, if the scissors cut is vertical, I think you can appreciate that the ends have now been exchanged. Now the big A black end goes with the little b red end, and the little a red end goes with the big B black end. So that's a chromosomal exchange between the two. And in addition, there's a little region in between where it's a red-black heteroduplex, which then can resolve just like we described. Okay, so those are the two fates of that holiday junction. And what this looks like, this is just redrawing it in sort of cartoon form. If we just ask about the fates of, and sort of restating what I just said, of a region in the middle that's heteroduplex, I think you can appreciate it can end up being corrected so that it's all the capital B black version. It could all be the all be the lowercase b, the red version, or it could be a mixture, red and black or black and red. Okay, all those are possible. Now, this is a big deal because this kind of recombination not just happens in meiosis. You'll hear about this in the genetics course if you're going to take that. There are a few recombinations, 2 to 3 per human chromosome per meiosis of this sort, and that's sort of a shuffling of the genetic deck of cards that makes each of us different from each other. But it also happens mitotically, that is in somatic cells, and that generally has no consequence because who cares whether, let's say, one arm of your father's chromosome and the corresponding arm of your mother's chromosome exchange with each other as long as those pieces are still present in the same cell at the same time. But if one, and here's where it can be trouble, if one of those chromosomes carries a mutation in a gene called a tumor suppressor, so these are a set of genes, there are several dozen in humans, which are basically, you can think of them as the brake pedals on a car. They keep cell proliferation under control. If you have one mutant copy and one good copy, you're basically fine, but you're at high risk for cancer. And BRCA1 is an example of one of these. If you inherit one mutant copy, you're at high risk for breast and ovarian cancer, or if you're a male for various, including actually breast cancer in males, various cancers. And the reason that you're at high risk is because a mitotic recombination between the chromosomes can lead to cell progeny that are homozygous for the mutant form. So let's just follow how that happens. Here are two versions of a single chromosome. Let's just say it's chromosome 7. I can't remember which chromosome this particular tumor suppressor RB happens to be on, but let's call it chromosome 7. Here's the mom's copy, the dad's copy. The dad's copy has a mutation, mom's copy is fine. The cell having one normal copy is still basically fine in terms of growth control. But now the chromosomes have just replicated. Now these perfect identical copies are from mom and from dad. And dad has the 2 mutant copies, mom has the 2 normal copies. But if one of these mitotic recombination events happens between these chromosome arms of the 2 different homologs, then I think you can see dad's arm with the mutant copy is now on this piece of mom's chromosome and vice versa on this one over here. And when the chromosomes separate, remember they're pulled by their centromeres to the 2 progeny, to the 2 daughter cells, one daughter gets, let's say, the left chromosome here, that's this one. The other daughter gets the left chromosome of this one, that's over here. And then the second daughter gets the 2 versions that have the mutation. So this daughter cell now has only the 2 mutant copies of the tumor suppressor. And that cell is now on its way to becoming a cancerous cell. So, of course, there are additional genetic changes that have to happen, but this is the first step. So this sort of mitotic recombination, although not that common, is also not that rare. And so that's why people who inherit one mutant copy of a tumor suppressor are at high risk for cancer. Okay, let's talk a little bit about radiation. This part's just kind of for fun. As you probably know from physics, we can only see a tiny sliver of the electromagnetic spectrum. Here it is. Red, orange, yellow, green, blue, violet. And to the longer wave side are infrared, microwaves, radio waves. These are lower energy, right? Longer wavelength, lower energy. Towards the shorter wave side, we get to ultraviolet, X-rays, and then gamma rays. This is higher and higher energy. And ultraviolet is a energy level that can make and break things like carbon-carbon or carbon-nitrogen bonds. And X-rays and gamma rays can actually fragment DNA. And it's interesting to look at resistance to very high energy radiation. We normally don't receive that much of it. There's some sort of constant level of cosmic radiation hitting us. It's not that great, but we can generate it artificially. And people discovered early on, I think this was back in the 1940s and 50s, when they started irradiating meat as a way of sterilizing it. So you may know in the meat packing business, one way of extending the shelf life of meat is to irradiate it. And they used X-rays and gamma rays. And what they found was that there was a particular species of bacterium that was amazingly resistant to radiation. So this was the discovery of this bug called Deinococcus radiodurans, like radiation durable. And there's another one, Thermococcus gamma tolerans. You can see how the names came, tolerant to gamma radiation. And just to give you an idea how radiation resistant these are, so the radiation from gamma rays and X-rays are measured in a unit called grays, GY, and a chest X-ray gives an exposure about 1 milligray, 1/1000th of a gray. 5 grays, 5,000 times that, is a lethal dose for us, and 500 grays is a lethal dose for E. coli. But Deinococcus radiodurans and Thermococcus gamma tolerans can survive 5,000 grays, okay, 10 times more than E. coli. It's probably not that they were selected to survive radiation, but it's probably a side effect of surviving desiccation. That's what people think. And desiccating a bacterium for an extended period is a stressor where there's genomic damage of the same sort or it can be repaired by the same mechanism that can repair radiation damage. And so people have studied what that repair system looks like. And here's a very simple experiment. This is a gel. This is gel electrophoresis of DNA, but it's a special kind of gel called a pulse field gel. Probably none of you have run any of these. They sort of had their moment of glory 10 or 20 years ago. Has anyone ever run a pulse field gel? Nobody. Well, I ran one long ago. Okay. So it basically resolves very high molecular weight DNA, and it does it by pulsing the electric field. First you have an electric field. Typically it's at 45 degrees. It goes from upper left to lower right for about a minute, and then you switch from upper right to lower left for a minute. And what that does is if you have a very long piece of DNA, it's like a big piece of spaghetti, it changes its shape in the gel as it gets extended by the electric field But then a minute later, it has to change its shape to a different direction and then change back and forth. And by that back and forth changing of shape, the DNA sieves differently through the gel depending on its molecular weight. As it turns out, very large molecular weight fragments can be separated. So the size standard that you see here, this isn't your usual size standard. These are lambda multimers. Lambda is 50,000 bases. So this is 50,000, 100,000, 150,000, 200,000, and so on. You can separate up to about 1 million bases in length. And this is the Deinococcus radiodurans genome, the whole bacterial genome, cut with a restriction enzyme, one that cuts very infrequently, recognizes 8 bases. And here are the fragments of the normal genome separated on this gel. There are about 10 of them. And then after irradiation, the same sample, same number of cells is run on the gel, and holy cow, first of all, a lot of the DNA is stuck at the origin, which it was anyway in the control, but the rest of the DNA that's in the gel is very low molecular weight. And yet these cells are going to survive. And if you look a half an hour later, it looks pretty much the same. An hour later, there's less of this low molecular weight stuff. There's more stuff at the origin, I can tell you. An hour and a half later, 2 hours later, but then 3 hours later, presto, the genome is back together again. What's happened? The genome has been smashed to smithereens by the gamma radiation. There's no way any bacterium or any organism can avoid that, because the radiation just does what it'll do. But then all the pieces are put back together by homologous recombination. And so this is a very potent homologous recombination system that can reassemble genomes. Okay, so just a little summary here of homologous recombination. It's initiated from single-strand or double-stranded breaks, occurs through the generation and resolution of holiday junctions, generates diversity through the creation of new combinations of pre-existing genetic variation, like meiotic recombination in us, and can cause loss of heterozygosity in somatic cells, and as a result, cancer. And let me just conclude with one final mini topic, which didn't fit into any of the other categories, and that is an endogenous mutagenic system. It's called ApoBec. It's a family of enzymes, and the ApoBecs are, and there are about a dozen of them in us. They're a series of enzymes that result in deamination of cytosine. They convert cytosine to uracil, and they can do it, depending which enzyme you look at, in RNA or DNA. And people think they're basically there as viral defense mechanisms. So if a cell is infected with a virus, the best thing the cell can do for the entire organism, if it's a multicellular organism like us, is to halt the viral infection and commit suicide. So it induces the ApoBec enzyme as an antiviral response. It causes lots of mutations in the viral genome. Of course, it causes lots of mutations in its own genome, and the cell kills itself, but no progeny virus emerge. But this turns out to be a very big deal in lots of non-infectious contexts, and in particular in cancer. So I think I just have this listed in words on the next slide. The ApoBecs, I'll just say what the name is. It stands for apolipoprotein B mRNA editing enzyme catalytic polypeptide. What a mouthful. But this was discovered because this enzyme that encodes apolipoprotein, the message that encodes apolipoprotein B was discovered to have a sequence, a single nucleotide, it's always the same nucleotide in the same place, that is different from the sequence encoded in the genome. That didn't make any sense. And then people realized there's an enzyme that comes along and finds that messenger RNA, finds that context, and mutates that base. And turns out that then encodes a different amino acid. Why nature does it that way, I have no idea, but it seems like a lot of work. But that was the original member of the family, but now, and the conversion is C to U, in this case in messenger RNA, but now we know there are other enzymes that convert C to U in DNA. As I mentioned, it's an antiviral defense. But now we also know that when you sequence cancer genomes, and thousands of these have been sequenced, and you compare the sequence to the normal genome sequence of the same person, let's say from their white blood cells, many of the mutations in many cancers are just the kind that you get from ApoBec-derived mutagenesis. And in fact, in many cancers, ApoBec enzymes, one or more, are highly upregulated. So there's this sort of pro-mutagenic mechanism going on in the cancer cells, which of course from a Darwinian survival point of view is probably a good strategy for the cancer cells. So this regulation of the apobex, which is normally very tight to keep them very tightly repressed unless there's an infection, is not perfect and can be taken advantage of by cancer cells. So just to sum up, since now it's been an hour and a quarter, we have in us about 10,000 oxidation events per cell per day, somewhere between 1,000 and 10,000 depurination events per cell per day, and then all these other events, alkylation events and so on, so that in the 10 to the 14 cells in our bodies, during this roughly 1 hour lecture, we have all had about 10 to the 17 DNA lesions. Hopefully all of them have been repaired. But 10 to the 17, that's even bigger than the national debt. So 10 to the 17 is 100 million billion. Okay, so there you have it. So you learned about repair and you've been doing repair. What more can you ask for? Thank you. I'd be happy to take any questions. Yes?
So the apobex system that you mentioned, so in cells normally it's tightly regulated, and in cancer cells they have mutations of the system, and that's what undoes that regulation?
I don't know the answer to that. So cancers have a lot of gene misregulation. I don't know the exact mechanism. Among other things, they have a lot of epigenetic misregulation. For example, patterns of methylation are altered. But I'm not sure what it is for the apobec enzyme induction.
So does loss of BRCA1 and 2 just lead to decreased homology-directed DNA repair, or zero homology-directed repair?
Yeah, the question is, does loss of BRCA1 or 2, the two famous breast cancer genes, lead to just a decrease in homology-directed repair or a complete loss? I don't know the answer to that. My guess is it's not a complete loss, because I think most of these systems have some redundancies built in. It's not all relying on a single pathway or a single protein. That's just my guess. Okay, thank you very much. Enjoy the rest of the day. Thank you for saving the day. Oh my goodness, that could have been a brief lecture.